Welcome to the cell signaling course. We have been discussing different types of receptors, different ways by which signaling can be communicated and it meets its goal. In few of the last classes, we saw some receptors which are soluble type. They are not membrane bound. They are either present in cytosol or in the nucleus. They are in the class of nuclear receptors. And invariably, they have this transcription factor activity. We will discuss in today's class that how these nuclear receptors work, as well as we will discuss strong signaling aspects which are regulated by co-receptors which are present on the cell surface. So these nuclear receptors, there are almost 50 such receptors which have been classified into 7 families, and within each family there are subgroups. So first family is called family 0, which is kind of orphan receptors and ligands are not known. Family 1 has 7 subgroups. This is the largest subfamily or largest family with maximum number of subgroups. Here the ligands which are known are like thyroid hormone, retinoic acid or vitamin D receptors. Further, the family 2, which has 5 subgroups, often the ligands are not known, but some of the known ligands are hepatocyte nuclear factor and the testicular and retinoid receptors. So 3 families, family 0, family 1, family 2. What you realize that family 0 and 2 have many unknown ligands and thus are orphan receptors. Family 3 has 3 subgroups. Mostly the ligands are estrogen, androgen, progesterone, glucocorticoids and mineralocorticoids, basically the steroid type ligands. And the family 4 has unsaturated fatty acids and certain growth factors as their ligand. Family 5 uses phospholipids as some of the ligands. Is phospholipid type. And then we have family 6, which eventually makes it family 7 because of family 0 as the start thing. The germ cell nuclear factor and the ligands are not known. So we have been seeing that how actual receptors which are bound to the cell surface, they respond to variety of signals, whether it is amine type signal, nucleotide type of signal or lipids and maybe certain metabolites, certain hormones and so on. So what is seen in case of a soluble receptor or the nuclear receptors, they also have wide variety of these signals working on them. General structural classification of these receptors is such that there are domain wise arrangement where N-terminal, if I refer that as domain 1, and then there is a in between there is a DNA binding domain. There is a small hinge region present over there, and the C-terminus has the ligand binding domain. So this is more or less common arrangement of domains in the nuclear receptors. These N-terminal domain, which is important for many activities, and the ligand binding C-terminal domain, they are referred as activation function domains. And the C-terminal domain is something which is used for dimerization. Very much like other receptors, even nuclear receptors work by forming dimers. And this ligand binding domain is critical for that. So here what happens that the arrangement, the folding of the individual nuclear receptor is like this, and through this domain 3, which is ligand binding domain, it forms the dimer. In some cases, it is possible that dimer is formed in the absence of ligand, but the receptor becomes active only when ligand binds to it. So the different families which have been or different types of these nuclear receptor, based on how they function, what they activate, they have been classified into 4 types. This particular type, here the receptor lies in the cytoplasm, and there it is inhibited by its binding to the heat shock proteins. And the ligand binding removes the heat shock protein, receptor moves into the nucleus, where it binds to DNA elements, and some of these DNA elements are called hormone response element, particularly in case where receptor is responding to a hormone, and these response elements are present as palindrome. So this is what is shown here, that your receptor is present in the cytosol, and what has been mentioned that this is generally bound to a HSP, which keeps it inactive. Ligand binding actually removes the HSP, and then it moves to the nucleus, where this is your hormone response element, which is present as palindrome. Once this binding of the receptor bound to ligand happens to these hormone response elements, transcription is activated. The type 2 receptors, they are generally nuclear receptors. They are not in cytosol, they are present in the nucleus, and they are inhibited by binding of a co-repressor molecule. These receptors are generally heterodimer, where they form the heterodimer with retinoid X receptor. Both are nuclear receptors, but they exist as a heterodimer, and they are present in the nucleus in inhibited state with the binding of co-repressor protein. When the ligand comes, small molecule as a ligand passes through the membrane and comes into the nuclear periphery or nuclear region, it binds to the receptor and then replaces. It binds to the receptor and then removes the repressor and replaces it with the co-activator molecule. Now with this co-activator molecule, it is bound to the hormone response element. Here it is not as a palindrome sequence, rather it is a direct repeat sequence, and then this binding activates the transcription. So in brief, what happens, that co-activator generally they are bound, they bound only when the ligand is present. Normally the co-repressor is bound to the receptor and they are present as a heterodimer. Here the HRE elements which are here, they are tandem repeat or direct repeats. When ligand comes in and it binds to the receptor, it replaces, removes co-repressor and replaces or binds with co-activator. This co-activator allows transcription. So co-repressor generally they have some deacetylase activity which blocks the transcription. That is why although the receptor is present inside the nucleus, it is inhibited. When ligand comes, removes co-repressor, it must place the co-activator which have acetylation activity towards histone, acetylation activity towards histones, which allows the transcription. The type 3 receptors, they are also present in the nucleus as a dimer, but as a homodimer. Rest everything remains same, that they are bound to the receptor and they are bound to the co-repressor and inactivated. Ligand binding removes co-repressor, puts the co-activator over there. Again the HRE element is a direct repeat which activates the transcription. So what happens in case of type 3 nuclear receptor? It is homodimeric situation. With the receptor, binding of ligand removes co-repressor, places co-activator, and the tandem repeat or direct repeat of HRE, where this binding of homodimeric receptor is happening, which allows the transcription. So what we have understood is that in the two cases of these nuclear receptor type 2 and type 3, the more or less mechanism of activation is same, by relieving the repression caused by co-repressor and activating the transcription by presence of co-activator. just that in case of type 2 it was heterodimeric receptor, now in case of type 3 it is homodimeric receptor. Type 4 receptor, which is the final type of nuclear receptors, they are almost same as type 3, just that the response element to which they are binding, they are not direct repeat, rather they are extended sequences which are present over there. So the situation will be same, it will be homodimer type of receptor, receptor will be bound with the co-repressor and kept in inactive mode. Ligand binding removes co-repressor, places co-activator, and this co-activator will be bound to the HRE elements which are in the form of extended half repeat. So what is situation here is that rather than having tandem repeats, you have the extended half repeat. Eventually ligand binding, placement of co-activator, transcription is turned on. So this kind of suggests that there are different nuclear receptors, there are different cytosolic receptors, they activate transcription. Largely they have the transcription factor role. We saw that their overall general organization had this DNA binding domain, which was helping in the process of transcription. Let's talk about another set of receptors which are in the family of co-receptor. As the name suggests, they will be assisting a type of receptor, but they become integral and important for ensuring the signaling goes in right direction. Also sometimes these co-receptors could be assisting the process of signaling, or sometimes they might be inhibiting the process. Basically they remain bound to the actual receptor and inhibit the signaling. For signaling to continue, the co-receptor must be removed. So they play a very important role. We have seen multiple cases and we will discuss that. These co-receptor come in different flavors. They have a type of co-receptor where it is completely transmembrane molecule, very much like other receptor. Then there are co-receptors which are concentrated in membrane domain with the GPI anchor. And then third type of co-receptors are proteoglycan co-receptor, where there is a protein core and the protein core is modified by glycosylamine glycan. So extracellular matrix in connective tissues is where these glycosylamine glycans are present, which will be modifying the protein. So there are three distinct classes we can say are available for co-receptors. Often these transmembrane receptors which are there, they undergo proteolytic cleavage. We have seen proteolytic cleavage happening for regular receptors also. If you recollect, it was for EPH-ephrin like receptors, it was for Notch delta type of receptor that extracellular domain was being removed by proteolytic cleavage, which is referred as ectodomain shedding in case of co-receptors. So this is the scenario where you have the transmembrane co-receptor with the extracellular domain. It should have a place where proteolytic digestion can happen. Then you have this GPI anchored receptors, which basically means that it will not be having any extension into the cytosol. Then you have this receptors which could be having extracellular domain which is modified with glycosaminoglycans which are present in extracellular matrix of connective tissue. This is some sort of regulation on the functioning of these receptors. So these receptors have been found in plenty and work through effectors and mediate the signaling. Independent of their receptor, sometimes they add to the functioning of the actual receptor. So there are co-receptor families like shown here: LRP, P75, NTR, Syndecan, interleukin, or TGF beta. There are some key effector molecule, you would have seen some of them coming in previous classes like SHC, PI3 kinase, RhoGDI, arrestin, SARC, alpha-actinin, or PIP2 binding, and so on. And basically each one of them, depending on what kind of receptor they are, what kind of effector molecule they are working through, they mediate certain signaling aspect and the processes like Wnt signaling, which is important for early developmental process. We have seen MAP kinase working exclusively in plants for lot of responses mediated downstream of receptor like kinases. Migration, proliferation, and so on, which we saw for receptor tyrosine kinase signaling in multiple cases, or cell adhesion, where we saw how the integrin kind of receptor worked along with the focal adhesion kinases. We also saw example of JNK working or NF-kappa B molecules working, which affect the angiogenesis, which affect the apoptosis. Also there are these co-receptor which work through angiogenesis, and in particularly in case of receptor serine threonine kinase, we saw that TGF beta type co-receptors, they worked with activin signaling as well as BMP signaling. And if you recollect, BMP signaling was playing very important role in bone morphogenesis, and activin had lot of role to play in cancer, infection, immunity, and so on. Further, there are these molecules which help in integrating the signal which is there because of the extracellular matrix and the other receptors. So you can consider that this class of co-receptors, when they are integrating the signaling between ECM and other receptor, they might be into the class of the proteoglycan co-receptors. There are multiple other pathways which these things work, and eventually because of their function, they help in the formation of something called morphogen gradient. Many insects, when they are developing, presence of a small molecule in a gradient available to different cells in a tissue helps them shape or acquire particular fate. Wnt is one of the molecule which is available as a gradient, and that drives the morphogenesis in the tissue. Something like this, if you consider a layer of cells and you have a gradient of, suppose, Wnt here, which is referred as morphogen. Basically something which is affecting the morphogenesis is a morphogen. So something which gets higher dose of Wnt will take one fate, something which gets the lower dose takes the other fate. If I say fate 1 and then fate 2. Then these co-receptor help in generation of soluble extracellular domain. I told that these receptors might have some of these receptors, particularly the ones which are transmembrane type co-receptors. This transmembrane domain outside in the extracellular domain region has this proteolytic cleavage site. Because of this digestion, this molecule will be free to move around, and that's where it will generate the extracellular domain. This might bind to something and ensure that signaling moves in the proper direction. Then it will help in the localization of the signaling, because if the receptor, if you think about this, a receptor is present here and it needs the co-receptor for activation or inhibition. So the co-receptor is in some sense localizing the signaling to a particular region of the tissue. Also, they can independently do the signaling of its cognate receptor. If suppose there is a TGF beta receptor it is ensuring, suppose it was ensuring the case of bone homeostasis. So the co-receptor which is here, it may work independently. If you recollect, we saw in case of G alpha beta gamma, G alpha was having its own signal, beta gamma also had its own signal. But if you consider them as a trimeric, heterotrimeric G protein, then this is a single entity. However, when it came to signaling, because of their dissociation, it signaled independently of the beta gamma subunit. Here also the co-receptor may assist the actual receptor in signaling, but it may induce a signaling of its own. Further, through its ability, particularly the receptors which have these proteoglycan modification and interaction with extracellular matrix, it will help in the cell adhesion process. The cells which are expressing such co-receptors, they will hold the cell in its place into the extracellular matrix or into the connective tissue to ensure that cell is localized to a particular place. And we understand that how this process is very important, because if cell loses adhesion, it will probably move to a new location, and which is the characteristic of epithelial to mesenchymal transition, often seen in metastasis. Then it helps in the orchestration of the signal, because it will ensure that certain important molecules which are interacting with them, they will be present over there. You can also think it from the perspective that there was a set of receptor which we saw in this case, which were having the GPI anchor. So all the molecule which is required for activation of the signaling, they could be tethered to this GPI region to form a cluster. So basically this co-receptor, through its GPI anchor, is helping in the recruitment of all required entities as a cluster for the signaling process. So they play a very critical role in many aspects of development, proliferation, migration, cell adhesion, and many more. So what we have understood in this class, that type 1 of co-receptors, they remain inhibited in cytosol. Their ligand binding makes them active, move into the nucleus and activate the transcription. Type 2 to 4, they are nuclear receptors present always in the nucleus. They are bound to the co-repressor, thus they cannot activate the transcription. Binding of ligand switches it to the co-activator form, where it can bind to the DNA elements where it was already present and facilitate the transcription. The thyroid, ecdysone hormone, androgen, and many more steroid molecule, they signal through the nuclear receptors. In case of co-receptors, there are multiple different types. The one which has the transmembrane type of arrangement has an ectodomain where it can be proteolytically cleaved to modulate the signaling process. Sometimes this free molecule, they serve as a competitor and do not allow the ligand to bind. So co-receptors, together we have seen the multiple different types. They play a very important role in several aspects of signaling, thus growth, development, and if I can take here and say it plays all very important role in growth, development, migration, adhesion, and many more things, which is integral for the proper functioning of the tissue and the homeostatic environment which is present in the individual or the animal. Thank you.